Hi, my name is Kavya Garg, and this is my presentation on the cellular basis of MASLD and insulin resistance. In this presentation, I will be going over my review paper project, which was about MASLD and its cellular mechanisms, as well as how it interacts with insulin resistance to cause MASLD. Table of contents. This table of contents shows what will be covered in my presentation. An introduction, the prevalence of the disease, liver anatomy, causes of the disease, cells and cytokines involved, as well as treatments and a conclusion. First of all, what is MASLD? MASLD stands for Metabolic Dysfunction Associated Steatotic Liver Disease. MASLD is a type of liver injury that affects approximately 30% of the world's population. The disease is characterized by the excess deposition of fatty acids, called triglycerides, into liver cells called hepatocytes. This figure here shows how a healthy liver can progress from being healthy into simple steatosis, then into MASH, which stands for Metabolic Dysfunction Associated Steatohepatitis, into cirrhosis, which is liver scarring, and finally into liver cancer, also called hepatocellular carcinoma. Prevalence. Understanding MASLD is important for many reasons. MASLD is highly prevalent and a widespread disease. It affects a large portion of the population, and if it worsens, it can become very dangerous. This figure shows how widespread MASLD is. MASLD is highly prevalent because even in parts of the world where its prevalence is the lowest, such as Western Europe and East Asia, still over a quarter of the population is affected by MASLD. This shows how prevalent the disease is and why it's important for people to understand the disease so that people can become more aware of it. So, before I explain what exactly MASLD is and how exactly it works, I will need to go over some liver anatomy. This picture shows the anatomy of the liver. On the left, we have the outside anatomy of the liver, and on the right, we have the interior vascular structure of the liver. So on the left here, we see the right lobe, the hepatic vein, the inferior vena cava, and the aorta, as well as the left lobe. Mostly with the liver, MASLD affects just all of the liver cells and liver tissues, so just understanding the shape of the liver and where it is and how it works in accordance with the other systems of the body is important. Now, moving on to causes of the disease, MASLD usually begins with the presence of other metabolic disorders. One key example of a metabolic disorder involved with MASLD is insulin resistance. Insulin resistance is one of the main causes of MASLD. Insulin resistance occurs when the cells in your body are less responsive to insulin and cannot take up glucose from the blood. This figure here shows how when lipolysis occurs due to caloric surplus, which is when a person is having overnutrition and eating too many calories, they can get lipolysis. And lipolysis is a metabolic process that breaks down triglycerides and into glycerol and free fatty acids. This breakdown of things into the fats gets them transferred to the liver, where the fatty acids are deposited into the liver in increasing amounts, thus causing MASLD. Another big cause of MASLD is gut microbial dysbiosis. So what is gut microbial dysbiosis? Gut microbial dysbiosis occurs when the health of your gut microbe is compromised. One common example we see of gut microbial dysbiosis in life is when people's gut bacteria is imbalanced and they're having trouble with digestion. Gut microbial dysbiosis can not only cause digestive issues, but also make it so that molecules from the diet or molecules from what people eat can flood into the liver, which causes MASLD. Now, here are some cells and cytokines involved in MASLD. First of all, we have hepatocytes. Hepatocytes are the most abundant cell type in the liver. MASLD develops when triglycerides or fatty acids are deposited into hepatocytes. This figure on the right shows some histology of the liver. Here we can see these white circles all over, which are not actually cells, but instead fat deposits inside of a hepatocyte. So, as stated earlier, MASLD occurs when these triglycerides are deposited into the hepatocytes. So this really just shows how the liver can look when a person has MASLD. Now, moving on to Kupffer cells. Kupffer cells are a really important part of the liver because they are liver macrophages. Due to this, they're part of the liver's defense system. Whenever the liver gets hurt by something like lipotoxicity, Kupffer cells will activate the immune response and heal the damage done by lipotoxicity. Now, lipotoxicity is when fat accumulation in the liver causes cells to die and not work properly, which shows how Kupffer cells are a really big part of trying to combat against MASLD. Now, this here is not actually a cell type, but instead a process that occurs all over the body. I will be explaining what this angiogenesis is prior to the next two cell types because angiogenesis is highly important to these cell types processes and how they contribute to the worsening of MASLD. So what is angiogenesis? Angiogenesis is a condition caused by differing cell interactions during MASLD development. What happens is that angiogenesis generally has to do with the development of new blood vessels. What occurs during MASLD is that chronic liver damage happens. This can cause one of two things: the activation of Kupffer cells or the activation of HSC, or hepatic stellate cells. First, looking at the left side of this diagram, we can see that the activation of Kupffer cells can cause hypoxia to occur in the blood. Hypoxia or hypoxemia is when blood does not have enough oxygen in it. And when this hypoxia is happening, the liver is not getting enough oxygenated blood. In order to solve this, angiogenesis occurs because it wants to create new blood vessels in the liver so that oxygenated blood can flow to the liver. However, the consequence of this is that normal hepatic vascular structure, or the normal structure of the veins in the liver, as we saw earlier, will get disturbed. And what this does is it actually causes fibrosis and liver scarring, which can be very bad for the liver. Additionally, if you look on this side, after Kupffer cells are activated, pro-inflammatory cytokines get secreted, which causes inflammation and therefore again leads to fibrosis. On the other side, we have hepatic stellate cells. When these hepatic stellate cells are released, they cause the recruitment of monocyte-derived macrophages, which will deposit ECM, again causing fibrosis. So basically what this really shows is that angiogenesis, the creation of new blood vessels, can be highly dangerous for the liver during MASLD. Now, moving on to endothelial cells. Endothelial cells are cells that provide a barrier between the blood and the rest of the body's tissues. So hepatic sinusoidal endothelial cells are responsible for causing angiogenesis during MASLD. And this can be harmful to the liver because angiogenesis causes inflammation and fibrosis, as we just saw. Okay, moving on to our next cell type, hepatic stellate cells. As was just seen in our diagram of angiogenesis causing fibrosis, hepatic stellate cells play a big role during angiogenesis in exacerbating or worsening liver disease to its later stages. However, the normal function of a hepatic stellate cell is to preserve liver health. In this diagram here, we can see that when the liver gets injured, hepatic stellate cells are activated. And these hepatic stellate cells can do the following things here. However, sometimes hepatic stellate cells will do inflammatory signaling, which can cause apoptosis, which is targeted cell death. In most cases, apoptosis, targeted cell death, can actually be positive because it can kill cells that might be harming the liver. But during MASLD's development, sometimes the positive reaction of apoptosis can actually become negative and start killing cells that are healthy. And it's because of this that hepatic stellate cells can exacerbate liver disease to later stages. Now, looking at pro-inflammatory cytokines. Pro-inflammatory cytokines, which you might have seen earlier in some of the diagrams, are a type of signaling molecule. These cytokines create inflammation in the body, and there are many types of them. Today I'm going to be talking about 4 pro-inflammatory cytokines which are highly involved with MASLD. First, looking here on the left, I have this picture of interleukin 6. Interleukin 6 is a type of cytokine that modulates the growth and activation of cells during the inflammatory and immune responses. On the right, we have C-reactive protein. Elevated levels of C-reactive protein can indicate insulin resistance, making them a possible biomarker or predictor for the development of MASLD. What all the pro-inflammatory cytokines here have in common, and as well as the ones I will just show, is that they are all possible predictors or markers that show that MASLD is being developed or has developed. TNF-alpha. TNF-alpha is a pro-inflammatory cytokine that stands for tumor necrosis factor A, and it is involved in systemic inflammation. Too much TNF-alpha can signal for inflammatory response in healthy cells. TNF-alpha signals for a cancerous cell to die. However, if the TNF-alpha has a negative impact, it can cause the inflammatory response in healthy cells in the liver, which shows how it can be negative. Next, I will be talking about interleukin-1 beta. Interleukin-1 beta's pathway is responsible for increased insulin resistance. It further worsens fatty liver disease. This figure on the right shows how it can cause chronic inflammation and therefore insulin resistance. This chronic inflammation can also cause beta cell dysfunction and death. Beta cells are cells that actually secrete and create insulin. So when these cells dysfunction or die, it does increase insulin resistance as a consequence, which in turn will increase MASLD. Here are some treatments for the disease. Traditional treatments for MASLD have historically included diet and exercise. However, advances have been made and are continuing to be made in terms of treating the disease. Next, I will show a figure that details some more of these treatments. This picture here shows how MASLD, also known as NAFLD, non-alcoholic fatty liver disease, has been treated in the past. Weight loss has been prescribed through diet and exercise, as in these lifestyle modifications. Additionally, bariatric surgery has been recommended, which does gastric bypass and helps to get rid of the fat in the liver. Here, we also have therapeutic approach, which usually involves medications. Insulin sensitizers are a big one, as well as acids and antioxidants like vitamin E. Lastly, when people are in end-stage liver failure, such as hepatocellular carcinoma, which is liver cancer, they might get liver transplantation. So here are some new treatments. One big treatment that is used for MASLD is semaglutide. Semaglutide is historically been seen so far with semaglutide injections, which are used for treating obesity as well as diabetes. Semaglutide has also been key in treating MASLD, with some of the pros of its usage for MASLD listed here. One pro of using semaglutide to treat MASLD is an improvement in liver enzymes, the reduction of stiffness in the liver, and the improvement of metabolic parameters in patients. A con of its use could be gastrointestinal adverse effects, as well as gallbladder-related diseases. Lastly, a new treatment for MASLD is ZSP1601. ZSP1601 is a pan-phosphodiesterase that is effective in treating MASLD. It targets ferroptosis, seen here, which prevents liver cell death. Conclusion. To conclude, MASLD has many causes. The complexity of the disease, as well as its high frequency and increasing death toll, underscored the need for more research to be done on MASLD. Thank you.